Hi everybody, today we are going to see about biological databases. Introduction. Bioinformatics is mainly concerned with biological databases and software tools written to manipulate these biological databases. Database means a logically coherent collection of related data with inherent meaning built for a certain application. Databases can be seen in everyday life, for example, a phone book of a phone numbers and name entries is a database. A CD containing a collection of songs is a database. It is a general repository of information or records to be processed by a program. Biological databases are large-sized, organized collections of scientific data and information generated by individuals regarding a particular biological aspects, grouped and well documented. They are usually associated with computerized software designed to update, query, and retrieve components of data stored within the system. There are different types of databases. One may characterize the available biological databases by several different properties. Here is a list to help you think about the various properties of a particular database. Type of data: nucleotide sequences, protein sequence patterns or motifs, macromolecular 3D structure, gene expression data, metabolic pathways. Databases are broadly classified as generalized database and specialized database. For example, DNA, protein, carbohydrates are databases which are generalized. Databases of express sequence tags (EST), genome survey sequences (GSS), single nucleotide polymorphism (SNPs), RNA database are some of the specialized databases. First, we'll see about generalized database. Generalized database contains sequence database and structural database. Sequence database are those that have the individual record as sequence of either nucleotides or amino acids. That is, they may be either nucleic acid database or protein sequence database. Structure database are the one that contains the individual records as biochemically solved structures of macromolecules. The nucleic acid database may be divided into primary database and secondary database or value-added database. Primary database contains the data in their original form, taken as such from the source. Examples include GenBank, DDBJ, PIR, and SwissProt. Secondary database or value-added database contain annotated data and information. Example: OMM, Online Mendelian Inheritance in Man, Gene and Clinical Data, GDB, Genome Database, which will contain human, and ProCite blocks. It will be having this protein motifs information. Specialized database. There are a number of specialized databases available in the net. Some examples of these databases are Express Sequence Tag, Genome Survey Sequence, Single Nucleotide Polymorphisms, Sequence Tag Sites. Exclusive DNA databases are also available for some of the organisms like E. coli, Saccharomyces cerevisiae, Oryza sativa, Homo sapiens. And specialized protein family databases, protein classification databases, structure database, pathway database, and microarray database are also available. The first category of nucleotide sequence database is further classified into four. They are International Nucleotide Sequence Database Collaboration, Coding and Non-Coding DNA, Gene Structure, Introns and Exons, Splice Sites, And fourth is the transcriptional regulator sites and transcription factors. Next, we will see about nucleic acid sequence database. Because of the high rate of data production and the need for researchers to have rapid access to data, public databases have become the major medium through which genome sequence data are published. The rate of growth of DNA database has been following an exponential trend with a doubling time now estimated to be 9 to 12 months. Three premier institutes are considered as the authority in the nucleotide sequence database. They are EMBL, European Molecular Biology Laboratory, NCBI, National Center for Biotechnology Information, and DDBJ, DNA Data Bank of Japan. They are primary nucleotide sequence database. These institutes have an international nucleotide sequence database collaboration under which they share nucleotide on daily basis. The nucleotide database have reached such large size that they are available in subdivisions that allow searches or downloads that are more limited and hence less time consuming. For example, GenBank has currently 17 subdivisions. Next, we will see about EMBL, Nucleotide Sequence Database. European Molecular Biology Laboratory Nucleotide Sequence Database is maintained by the European Molecular Biology Laboratory at the European Bioinformatics Institute. It is a primary nucleotide resource in Europe. The EMBL Nucleotide Sequence Database is a comprehensive database of DNA and RNA sequence collected from the scientific literature and patent applications. It also includes directly submitted sequences from the researchers and sequencing groups. Data collection is done in collaboration with GenBank USA and the DNA Database of Japan (DDBJ). Next, we'll see about GenBank, that is Genetic Sequence Data Bank, is one of the fastest growing repositories of known genetic sequence. It is maintained by NCBI, National Center for Biotechnology Information of the National Library of Medicine, USA. It has a flat file structure that is an ASCII text file readable by both humans and computers. In addition to sequence data, GenBank files contain information like accession numbers and gene names, phylogenetic classification, and references to the published literature. And this is the website of the GenBank, www.ncbi.nlm.nih.gov. Next one is the DDBJ, DNA Data Bank of Japan. The DNA Data Bank of Japan is an international database of nucleotide sequence started in the year 1986. It is located in the National Institute of Genetics, Japan. Its website is www.ddbj.nig.ac.gp. Next, we'll see about structure databases. These are databases that contain a collection of structures of biological macromolecules. One of the best structure databases is the Worldwide Protein Data Bank. It consists of organizations that act as deposition, data processing, and distribution centers for PDB data. The founding members are the RCSB PDB from US and PDB Europe and PDBJ from Japan. So this BMRB, that is in the US group, joined the Worldwide Protein Data Bank in 2006. The mission of the Worldwide Protein Data Bank is to maintain a single protein data bank archive of macromolecular structural data that is freely and publicly available to the global community. The Worldwide Protein Data Bank is a single global archive for information about the 3D structure of biomacromolecules and their complexes as determined by X-ray crystallography, NMR spectroscopy, and cryo-electron microscopy. This site provides information about service provided by the individual member of organization and about projects undertaken by the Worldwide Protein Data Bank. As on December 2010, more than 70,000 structures of macromolecules are available in Worldwide Protein Data Bank. The Research Collaboratory for Structural Bioinformatics (PDB) curates and annotates PDB data. It also provides a variety of tools and resources. Users can perform simple and advanced searches based on annotations relating to sequence, structure, and function. These molecules are visualized, downloaded, and analyzed by users who range from students to specialized scientists. EMBL EBI's Protein Data Bank in Europe. It is a European resource for the collection, organization, and dissemination of data on biological macromolecular structure. Until January 2009, the Protein Data Bank in the Europe was known as the Macromolecular Structure Database. Next is the PDBJ, Protein Data Bank of Japan, which maintains a centralized PDB archives of macromolecular structure and provides integrated tools in collaboration with the RCSB, the BMRB in USA, and the PDB in Europe. Hope you have gained a knowledge regarding the biological database and where it is located and the basic information related to the nucleic acid sequence database and also about the structure database. Thank you for your patience listening. Thank you.